In villages across West Africa, there's a shrub that grows almost everywhere, along roadsides, in savanna grasslands, on the edges of farmland. Most people who pass it wouldn't give it a second look, but for generations, traditional healers have turned to this plant for one condition in particular: diabetes. Its name is Bridelllia ferruginea, and over the past 2 decades, it's quietly become one of the more heavily studied medicinal plants in West African pharmacological research, not because of marketing, but because scientists kept noticing the same traditional claims showing up again and again across different communities and different countries. So today, we're asking our usual question. Not, does this ancient plant cure diabetes? Not, is this just folklore? But, what does the actual research say? Let's start with the plant itself. What is Bridelllia ferruginea? Bridelllia ferruginea is a shrub or small tree, part of the Euphorbiaceae family, commonly found across tropical West Africa, in countries like Nigeria, Ghana, and Togo. It can grow up to around 45 feet tall in the right conditions, though it's more often seen as a smaller shrub along roadsides and in open grassland. In traditional medicine, it's not a one-condition plant. It's been used for arthritis, dysentery, constipation, skin conditions, oral infections, and even as an antidote for arrow poison in some historical accounts. But across multiple regions and multiple independent traditions, one use shows up consistently: chronic diabetes. That consistency is exactly the kind of pattern that gets a plant noticed by researchers. When completely separate communities without contact with each other arrive at the same traditional use for the same condition, it raises a legitimate scientific question. Is there something biologic? This is where it gets specific. Because different parts of the plant have been used in different regions, and that matters for both the folklore and the science. In Togolese traditional medicine, it's primarily the root bark that has historically been used for diabetes. In Ghana and Nigeria, the leaves are the most commonly used part, dried and prepared as a tea or decoction. In fact, researchers at Ghana's Center for Plant Medicine Research developed a standardized product they called Bridelllia tea, made specifically from the dried leaves, so they could study it more rigorously in a clinical setting. We'll come back to what that study actually found, because it's one of the most important and most honest parts of this whole story. The stem bark has also been studied separately, mostly for its antioxidant properties. So, depending on where you look, you'll find leaves, root bark, or stem bark all described as the part used. That's not a contradiction. It reflects genuinely different regional traditions that developed somewhat independently, all converging on the same plant for the same purpose. For research purposes, the leaves are by far the most extensively studied part when it comes specifically to blood sugar effects. What's actually in the plant? When researchers ran phytochemical screening on Bridelllia ferruginea extracts, they found a fairly rich mix of compounds: tannins, flavonoids, saponins, terpenoids, alkaloids, and catechins. A few of these are worth knowing by name because they show up repeatedly in the research explaining why this plant might do something biologically. Catechins, a type of flavonoid also found in green tea, have been specifically identified in the root bark and studied as potential anti-diabetic compounds. Tannins, concentrated and isolated from the leaves in more recent studies, have shown stronger blood sugar-related effects in animal models than the raw leaf extract alone, which is part of why some researchers have tried to isolate and concentrate that specific fraction rather than using the whole leaf. None of this proves effectiveness in humans on its own, but it does explain why researchers didn't just dismiss the traditional claim outright. There are real, biologically active compounds in this plant, and some of them have documented effects on the exact systems involved in blood sugar regulation. So what's the actual proposed mechanism? Traditional practitioners wouldn't have described it this way, but modern research has investigated a few overlapping pathways. Enzyme inhibition. Some extracts from Bridelllia ferruginea, particularly from the flowers and leaves, have shown the ability to inhibit two enzymes called alpha-glucosidase and alpha-amylase. These are the same enzymes targeted by a real prescription diabetes medication called acarbose. In laboratory testing, some Bridelllia extracts inhibited these enzymes with a potency comparable to, or in some specific extracts, even stronger than, acarbose itself. This matters because these enzymes are involved in breaking down carbohydrates into glucose in the digestive system. Slowing that process can blunt blood sugar spikes after eating. Antioxidant activity. Multiple studies have found that Bridelllia extracts can scavenge free radicals in laboratory testing, reducing oxidative stress markers. This matters for diabetes specifically because chronic high blood sugar generates significant oxidative damage throughout the body, including to the pancreas, kidneys, and blood vessels. An extract that reduces that oxidative burden could, in theory, help limit some diabetes-related tissue damage, independent of any direct effect on blood sugar itself. Insulin signaling and glucose uptake. In one 2021 study on diabetic rats, a butanol fraction of Bridelllia ferruginea leaves was associated with upregulation of GLUT4, a transporter protein responsible for moving glucose out of the bloodstream and into cells. Researchers also found improvements in insulin signaling pathways in liver tissue in that same study. Kidney protection. In streptozotocin-induced diabetic rats, a standard lab model for studying diabetes, Bridelllia ferruginea leaf fractions were found to reduce markers of kidney damage associated with diabetic nephropathy, one of the more serious long-term complications of poorly controlled diabetes. Put together, the proposed picture is a plant that may work on multiple fronts at once, slowing carbohydrate breakdown, reducing oxidative stress, and possibly supporting insulin signaling and glucose transport. That's a genuinely interesting pharmacological profile. But interesting mechanism in a rat study and proven treatment for a human being are two very different things. And this is exactly where we need to slow down. What the evidence actually shows. Here's the honest breakdown. The overwhelming majority of research on Bridelllia ferruginea and diabetes has been conducted in animals, specifically rats and mice with chemically induced diabetes, or in laboratory test tubes measuring enzyme activity. Study after study across different research groups in Togo, Ghana, and Nigeria has found statistically significant reductions in blood glucose, improvements in cholesterol and triglyceride levels, and reduced markers of kidney and liver damage in rodents. That's a genuinely consistent pattern across independent labs, which is scientifically meaningful. It's not one study getting lucky. It's a real, repeated signal in animal models. But here's the part that often gets left out of the more excited corners of the internet. There's been very little rigorous human research. In fact, there's really only one clinical study that looked at this directly in real patients, and its results were far more modest than the animal data would suggest. Researchers at Ghana's Center for Plant Medicine Research conducted a retrospective clinical study on their standardized Bridelllia tea made from dried leaves. They reviewed records of 22 diabetic patients who had used the tea. The result? Fasting blood sugar did decrease slightly on average, but the researchers themselves concluded that the reduction was not statistically or clinically significant. In their own words, they noted the need to review the product further. That's an important, honest data point. It doesn't mean the plant does nothing. A small retrospective study of 22 uncontrolled patient records is a weak study design, and it can easily miss a real but modest effect or be confounded by other factors like diet, other medications, or how consistently people actually drank the tea. But it also means we cannot currently say, based on real human evidence, that Bridelllia ferruginea is an effective treatment for diabetes. What we can say is that it's biologically active, mechanistically plausible, and consistently effective in animal models, which makes it a legitimate candidate for further, better-designed human research. That's a very different, much more honest claim than this ancient remedy controls blood sugar. This matters just as much as the effectiveness question. There is no established, clinically validated human dose for Bridelllia ferruginea. The animal studies use doses calculated per kilogram of body weight in rats. Those numbers don't translate directly or safely to a human dose, and no regulatory body has set a verified therapeutic dose for people. We also don't have solid data on how Bridelllia interacts with actual diabetes medications like metformin or insulin. One study title we found even referenced reevaluating the leaf extract's use in combination with metformin, which tells us researchers themselves recognize this is an open, unresolved question, not a settled one. Herbal products in general, and this has come up before on this channel, can vary enormously in potency and purity depending on how they're grown, harvested, and prepared. A product labeled Bridelllia from one source is not guaranteed to match another in concentration or safety. So here is the honest, responsible summary. This is a plant worth continued scientific interest. It is not a verified, dosed, regulated treatment for diabetes in humans. Anyone managing diabetes who is curious about it should raise it directly with their doctor, not as a replacement for their current treatment, but as a question about whether, how, and if it could safely fit alongside it, given their specific medications and health history. The bigger picture. This is the same pattern we keep finding on this channel across very different traditions and very different continents. A plant gets used for generations based on observation, not laboratory science. Modern researchers investigate it and find real biologically plausible mechanisms, not nothing, not magic, but something. Animal studies show a consistent, real effect. And then, when it finally gets tested properly in actual human beings, the results are more modest, more uncertain, and more complicated than the traditional claim or the animal data alone would suggest. That gap between promising lab research and proven human treatment is exactly where most miracle plant claims online quietly skip a step. They'll cite the rat study. They won't mention the one human study that came back underwhelming. We're not going to do that here. Bridelllia ferruginea is a legitimately interesting plant, backed by consistent animal research and identifiable active compounds with plausible mechanisms. It deserves more better-funded human clinical trials, not because we're certain it works, but because the current evidence is genuinely promising enough to justify finding out properly. Until that research exists, it stays exactly where it belongs. An area of active scientific interest, not a verified treatment. If you or someone you love is managing diabetes and curious about a plant like this, here's the one piece of advice worth taking from this entire video. Bring it to your doctor. Ask what's actually been studied. Ask what's known about interactions with your specific medications, and don't change your treatment based on a single animal study or a headline. That conversation will protect you more than any plant, ancient or modern, ever could on its own. If you found this useful, subscribe, because we're going to keep doing exactly this. Taking traditional remedies people are genuinely curious about and giving you the real answer, not the clickbait one. Until next time, take care of your body, question what you hear, and never stop learning.